Thank you for the opportunity to talk to you today. My background is particularly in early stage clinical development, so that's where I'm going to focus most of what I talk about today, but I will give some pieces around how that is affecting the way that we approach large-scale clinical trials. I hope much of this is familiar to you about why do we need drug development to understand whether some funky science that we're doing is actually going to be a useful medicine. But I think this is particularly important because I think one of the critical lessons of the last 10 years is very often funky science does something that ends up looking very like the things that we've got already and doesn't necessarily, just because it's novel, just because it's a different mechanism, doesn't mean that the clinical benefit that patients are going to derive from that is going to be different from simple things that are around already. And so that you need to bear that in mind right at the start of drug development. I think it's one of those things that's always a bit strange when people criticize the magnitude of the treatment effect associated with new drugs. But just think for a moment, do you think anybody ever sets off down this path to try to develop a marginal improvement? Everybody starts off with the expectation and the hope that what you're developing is something that's going to make a real difference. But what often happens during the course of this is that you identify that that change isn't going to be as big as you think. And I think too often in the past, that has led to things being pushed through the development path. But I think what we will see in the future now is because of the payer environment, those things will actually get stopped rather than going forward. This is too small to read, and it's deliberately too small to read because the key point is this is the chevron diagram that everyone is taught about the way drug development is done. And it suggests that this is a linear process and it's just a series of things that you do in order to get to the final point of submission and marketing. And what I want to suggest to you today particularly is it's much more than that. What it really is a series of questions that you need the answers to move through this, and as a result it looks very different in 2014, 2015 than it did when these things were originally developed. When you're thinking about developing a new medicine, you need to think about where you're going. And again, this comes back to some points that Martin was making before. So when many of the targets that we look at these days have many potential indications, and I'll show you a specific example of that later on. What you need to think about right from the start is if it could work for heart attack, ingrowing toenail, psoriasis, all of these potential things, because the biology suggests that those things are important. Which one of those things is the thing where the scientific link is the most important? Because that is the area where you've got the greatest chance of being able to show the clinical benefit. But you need to think about that right from the start. And then you need to think about how that is going to link through to the different patient populations that you want to treat. And this comes back to this critical point. When you're in large-scale clinical trials, you can really only answer one question. And if you don't answer it in the right patient population or the right patient subpopulation within a disease, then you're not going to put yourself in a good position to have that discussion with payers, regulators, and those prescribers to actually understand the value of your medicine. And a critical thing that we face these days is, what is this medicine offering that is really different from the things that are available at the moment? There are now relatively few conditions where there is absolutely no treatment available to patients. So in the majority of cases, you're looking for something. Is it really going to make a difference in this space, or is it just a different flavour or different option? Because the truth is, these days, you could easily end up with a safe and effective medicine that nobody wants to buy. And as a commercial entity, that might well end in the closure of your company, because you've spent all of the money, which is hundreds of millions of pounds at that point, and you've got nothing to show for it. And you need to think about how the needs of these different groups feeds into what the data and the evidence that you're going to generate. The payers, the prescribers, and the regulators have different needs and different types of information that they're looking for, and you have to address all of them. But you can't necessarily address all of those questions in a single entity. Your randomized controlled trial may not give the payers the kinds of data that they're looking for, but you need to think about how you're going to build that evidence base. And you need to think about it right from the start, because it may well be that patients who are randomized to your very earliest studies, if you bother to follow them up, you could actually have 5-year data by the time that you come to registration. It may not be an enormous data set, but it's going to be better than 3-month data in a few thousand people in some cases. So where do drugs come from? Again, a lot of this will be familiar to you, but I just want to highlight particularly this final one. We've had an explosion in the last few years of genetic data and other systems biology data. The real challenge for us in drug development these days is really understanding what that means, and is this important in the pathological pathway of this disease, and is there redundancy in that system? And that's one of the things that you will see a particular focus on these days. I think you will see a reduction in the number of targets that pharmaceutical companies and others interested in drug development are trying to do. But what we're trying to do is improve the quality of those targets. And I'll come on to the reasons why that's been a particular problem in a moment. So in terms of an early clinical plan, there's some stuff that you know before you go into the clinic, but it's not very much, to be honest. You know some chemical information about the drug and a little bit about its formulation. You may have some in vivo pharmacology and in vitro pharmacology. You may have some genetic information. You've got some short-term toxicology that suggests in an animal species it's not too toxic. And you might have a little bit of information about the drug disposition, ADME characteristics of the drug. That doesn't actually tell you very much about what you really want to know as a drug developer. It's the platform that you have, but it doesn't actually address the questions that you need to answer. And what you really want to know as a drug developer is the drug sufficiently safe and tolerable? And clearly that's going to vary a lot depending on the condition and the background that you're treating. And can I give it for long enough for it to show a clinical benefit? 28-day toxicology will allow you to dose up to a month. Is that long enough for a disease-modifying treatment, that's going to affect joint integrity or something like that? Because to be honest, if it isn't, then you're not going to be able to answer those questions in your early clinical studies. So you're going to be carrying that risk forward, and you need to think about what that means in terms of your drug development plans. And the other critical question is really, is the drug engaging with its target for long enough to have a potential clinical benefit? And have you actually measured that, or have you just measured the concentration of the drug in the blood and hoped that that will tell you something about the concentration of the drug at its target? Because the majority of the targets these days are not in the blood. So when you're thinking about these early types of studies, you do need to think about where you're going. Because if you design the study based on what is easy and straightforward to do, that may not get you to where you need to be ultimately in order to support those large phase 3 clinical studies. So you need to think of this in two terms. One, about the disease that I'm trying to study. Is it acute? Is it chronic? What is the natural history? And this is very often an area where actually there's a big gap in terms of the understanding. What is the natural course of the disease in the population that I'm going to be studying? There may be epidemiological data about people with kidney disease in general, but is there the detailed information on the subpopulation that you are planning to study? Do you know the rate of deterioration of their renal function? Because if you don't, how on earth are you going to tell whether your drug is having an effect? And certainly, from my perspective, this is very often where we have a gap. And very often we need to invest in collecting this information before we get into the clinic. What about the population? Is it a young versus elderly population? The vast majority of drugs will be targeted at older people, and these days very often very much older people. So again, if you do your clinical studies in people in their 60s, but actually your target patient population in their 80s, is that really any different from doing the studies in healthy volunteers? What about comorbidities? Reducing comorbidities in your early clinical studies will make your life simpler, but does it mean that the data are then relevant? Do you in fact save now, but cause yourself problems down the line? Then what about the drug? You need to understand various things about the drug. And there is a temptation to try to answer all of these questions. All of these questions are quite easy to answer, and you can feel as if you're making progress. But what you're doing is generating data. If you don't know what questions you're trying to answer by generating these things, then you won't know where you want to go. And that's why I want to suggest to you, you need a question-based approach is helpful in early drug development. This is a traditional approach to early drug development, and certainly when I was taught about early drug development, this is the way things generally looked. You would do a first-into-human study, which would traditionally be in healthy male volunteers. You would then go on to, that would be a single-dose study. Then you'd go and give it over repeated doses, particularly focusing on pharmacokinetics. You might then go on and understand the drug disposition characteristics of that drug in more detail with a radio-labeled study. You'd then look at some different formulations, maybe powders, maybe different tablets. Look at the food effect of food. In parallel, you might be doing a safety study in patients just to make sure that the tolerability is acceptable. You may do some supporting drug interaction studies, all with the aim of getting you a profile of safety and tolerability in your patient population before going into your proof of concept study. And it's easy to criticize that type of approach because it's an awful lot of activity to get to that proof of concept study. But actually, it works very, very well as a concept if you know where you're going and you have a high probability of success that your mechanism is going to work. Because what it generates is a lot of information about your drug, and you can therefore have a high level of confidence as you go into that proof of concept study that you've got all of the information and you've designed it quite correctly. And the fact is, for precedented mechanisms, it's a great way of doing things. The problem is most of us are no longer working on precedented mechanisms, and again, the market for Me Too medicines has all but collapsed. It's slow, it's expensive, and it's not well applied to novel mechanisms. So if we look at a specific example here, the P38 MAP kinase, central second messenger system. Again, this is one of those examples. These are all ones I pulled out of the literature as potential targets for this drug. Huge variation there in different clinical conditions with different natural histories. This is not my area of work, but one of the critical things you'd need to work out in this space is from that smorgasbord of potential indications, which of these really has the the best science linking it through. And this is one of the real problems with a lot of the modern targets, is they could work for loads of things, but they probably don't work for anything. So how are you going to find that out? And that's why you really need to focus on things like, does the drug hit the target? And crucially, if it's hitting the target, is it having the downstream effects that you think in a later stage are going to translate into that clinical benefit? Because all too often we see these days is you engage the target, you get the primary pharmacology that you want, but because there are other mechanisms in play, you don't see those downstream effects. So if you focus entirely on pharmacology, that's great, but if you don't, you need those downstream effects as well. So what does early drug development look like these days? Very often, our first into human study will include a pharmacodynamic, probably a pharmacology marker. That may or may not be straight into patients these days. It may be simpler to do it in healthy volunteers, and I have nothing against doing studies in healthy volunteers if the target is there and that you can usefully use that information to get you into that patient population to really understand what your pharmacodynamic marker is, and crucially some downstream markers as well, and then moving through quickly into a proof of concept study. So this is good about quickly establishing about whether you've potentially got a developable drug and establishing that information around the downstream effect. The real problem, however, is that the studies to do this are very complex. We were hearing earlier about the importance in late-stage development of asking simple questions and applying them simply. The truth is here, the map is not worked out. You have to include lots of things to understand where you're going, understand the concept. Very often you'll have complex imaging as part of your study. And any of you who've done that will know that that adds substantial cost. So it's not uncommon for these kinds of studies to cost £15,000 to £20,000 a patient to evaluate. You're also going to be evaluating quite a small number of people. You'll get a lot of rich data from those people, but when you wrap up your safety database at the end of this kind of development, you may have exposed 25, maybe 50 people. That's going to be very different from taking a traditional approach where at the same stage you may have exposed several hundred other people. You may have better data, but you still don't have many numbers. And therefore you are carrying things, particularly around safety, later into development. And the other critical thing here is you're absolutely dependent on those pharmacodynamic markers, because that's your compass to know whether you're going. And if your PD marker is no good, then you will be going off in the wrong direction. And this is why it's so critically important. I alluded to this earlier. This is a relatively recent Nature review of drug discovery. Over half of drugs now fail in phase 2 because they don't work, or they don't work enough to be useful. If you look at those things that traditionally stopped drugs from being developed, particularly pharmacokinetics and bioavailability, actually our chemists are really good these days. It is very unusual to be going forward into the clinic these days with brick dust that is completely insoluble and doesn't have developable characteristics. The most likely reason you'll fail is because your mechanism is not as important as you thought it was in the disease that you're studying. And that's why you've absolutely got to focus on that at the very earliest stage of your drug development. When we think, if you manage to get through phase 2 and you've got some positive pharmacodynamic data and you may have some preliminary clinical data, in the past we then moved from this learning phase into the confirming phase. So if I just give an example here of an H2 antagonist, the phase 2b studies with an H2 antagonist were smaller versions of the phase 3 study. You did a range of doses, you gave them, people with a stomach ulcer or a peptic ulcer, you did an endoscopy at the beginning and the end and you looked for ulcer healing. What you did in phase 3 was do that on a much bigger scale. The translational risk between those phases is very low. I'm looking for, I know it out of phase 2b where I'm learning, and I'm pretty sure of what magnitude of treatment effect I'm going to see when I go into my phase 3. So it's relatively low risk. The challenge, however, these days is because we're with novel mechanisms, very often you've got pharmacodynamic markers, but not necessarily clinical endpoints in your phase 2 studies. You're actually moving from a learning phase into a still learning phase. And I just got some examples here. You know, there's a famous one from GSK recently where we spent, we did 2 large 20,000 patient studies looking at daraplidib, particularly around atherosclerotic plaque volume. Very positive data in phase 2, but when you move into phase 3, then you're looking at these clinical endpoints, and the fact is it didn't translate in this case. So that's been published now, and it didn't translate. Huge investment, not simply of money, but resource and time to get to those questions, because the only way you can answer those questions is with very, very large studies. So about 40,000 people enrolled in those studies. Other ones are slightly better because we do understand the translational relationship slightly better. You know, the translational risk around glycemic control and microvascular complications isn't such a great risk. If you've got very clear glycemic control out of your phase 2 studies, you can be reasonably confident that you're going to see those changes. But very often for novel mechanisms, you don't have that. And again, this was alluded to as well. You have this additional complication of the fact that you want to be doing these studies when you're making that kind of investment to support global registration. So have you got the right populations? Have you got the right mix of populations? What about the different standards of care in those different geographies? And how do you manage risk going forward? Because again, going into these studies, you probably have got less safety data than you would have had if you'd taken a traditional approach. How are you going to manage that? So I think what you will see is much more of this, rather than this learning confirming, is many more trials now have this much more feedback throughout them. This is where, interim analyses, different looks throughout the study to see, are we on track to where we're going? Because you can't be as certain as you would have been with a traditional approach. I think we're also seeing that now changing in terms of the way that the regulators approach this. So here is, on the top there is the traditional scheme. You've got your clinical development package, you put it all together, you present it to the regulators, they ask you questions, you have a dialogue about those questions, and then you're given a marketing authorization. And then your interactions in the future with the regulators are really around any safety signals that might emerge, but also when you then go back to look for additional indications and new formulations. But there's an inherent weakness in this approach. It assumes that you pretty much know what's going on at that point of marketing authorization. And that's a real weakness in the system, because I think we all recognize that there are still a very large number of questions that may not have been answered. And there are now some schemes, particularly adaptive licensing, that the EMA are piloting at the moment. There are different schemes, but the key point is this idea that new medicines may be brought to subgroups or particular populations in whom you have demonstrated a benefit risk, but with an ongoing commitment to follow those patients up in a rigorous fashion to get those long-term data that are so often missing from drug development packages, with the idea that this will be more of a loop, just as we've seen in the clinical trials. It's more of a loop than it used to be, that there will be more of a loop with the regulators and more of a building of the clinical trial package, rather than you do it all, package it up and send it off approach. I just want to finish with just a few points around good clinical practice, because I think this is something where, certainly in conversation with people, I think there can be a little bit of confusion and about how this fits in. It's worth saying that, you know, good clinical practice came out of a need, particularly from the pharmaceutical industry, to have a common standard that would work whether you were trying to do register a drug in Europe, in the United States or Japan, which remain the largest markets for medicines in the world, although that is changing. And therefore, it came from that. And if you look at the parties who are involved in generating this, and this is under the umbrella of the International Conference on Harmonization, what you have is the industry body and the regulatory body from those areas. But it is worth bearing in mind is that is the driver for it. And most people think entirely in terms of good clinical practice, but it's worth saying that the ICH framework includes specific guidances on a very wide range of other things to do with drug development. Quality topics, particularly your colleagues who will be making the drug product for you will be intimately familiar with these things. What about stability testing? What about impurity testing? There are guidances on those. And then there are also safety topics, particularly about the non-clinical studies that you need to conduct. ICH is actually E6. It's actually just one of the guidances within the efficacy topics, which are the clinical ones. And it is worth being aware of these things, because if in your academic work or you're trying to do a study, you may well find that there's some very specific guidance that will help you on a lot of these other topics. And then finally, there are these multidisciplinary ones, and the ones you'll probably be most familiar with is this is where the dictionary that most of us use for classifying adverse events came from. So it is a whole group of things and a whole group of guidances. It's not just the GCP guidance. And again, this is too small to read, but deliberately so. One of the things that people often get hung up about is what I am doing a clinical trial under the aegises of the European Clinical Trials Directive. This is the flow sheet from the MHRA, but there are other flow sheets available as well. So I would encourage you to use those before you get a long way down the planning path. If you're thinking about a clinical study, try and make sure of that very early in the process so that you're absolutely clear what regulatory and other packages you need to get together, because I do sometimes see this from people. They've done a lot of work, they're really nearly ready to go, and then they work out that they fall under the Clinical Trials Directive, and then they're really put back a long way. They are very helpful, these flow sheets. There are a number of them available now. The other thing to say is the MHRA are very helpful, and they do have an inquiries line if you're not sure, because there are some of these things, and I think it's particularly true for those of you who are working in the academic field, is what I am doing a clinical trial under the directive or not. So just to try and summarize this piece, drug development is not that chevron diagrams of just I do this, I do this, I do this to generate data. Yes, you do generate data, but you must generate data to try to answer questions. And you need to be clear what those questions would be. You need to have that line of sight, but you also need to be realistic about what question can I answer at this stage, and what am I carrying forward into the next stage, because that's critical in terms of understanding your risk. If I show this, how much will this get me further forward in my drug development path? the whole framework for the conduct of clinical trials is changing, and now we're beginning to see the regulatory framework evolve in parallel with that. And just to remind you around the requirements that ICH GCP places on all of us that are conducting clinical trials.